Hello dear friends, I'm Gauri Suman from MD College, Parel. Today we'll be learning about cell division. This is the second presentation on meiosis. In the first video, we saw how to make smear slide to observe stages of meiosis. Today we'll learn about different stages of meiosis. Initially, observe the animated amoeba sisters clipping showing difference between two cell divisions, mitosis on the left and meiosis on the right. What have you observed? Yes. In mitosis, one cell divides into two daughter cells and they have the same number of chromosomes as that of the parent cell. Please observe carefully on the left. One cell divides into two cells. Whereas in meiosis, on your right hand side, one parent cell divides into four daughter cells having half the number of chromosomes as that present in the parent cell. Now we move on to a animated clipping showing different stages of meiosis. One cell divides into four cells. Please observe carefully. Okay, we move on now to definition of meiosis. It is a process of cell division. It occurs in reproductive or germ cells. It is not a continuous process like mitosis. It takes place only during gametogenesis in diploid cells. It is a very long process divided into meiosis 1 and meiosis 2. Meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. Prophase 1 is a complex process divided into 5 sub-stages, namely leptotein. Cell grows in size as a result of synthesis of DNA and RNA and proteins. The chromosomes appear long and distinct. Point to remember, chromosomes appear long and distinct. I repeat, chromosomes appear long and distinct. Zygotine, chromosomes become shorter and clear. Homologous chromosomes pair with each other. This is called as synapsis. The chromosomes are in bivalent stage. Point to remember, homologous chromosomes pair with each other. I repeat, homologous chromosomes pair with each other. Pachytene, the paired chromosomes become shorter and thicker. Each bivalent replicates to become tetravalent. Point to remember, bivalent becomes tetravalent. I repeat, bivalent becomes tetravalent. Diplotein, non-sister chromatids cross with each other. Due to this, different shapes are produced which are called as chiasmata. This is called as genetic exchange. Point to remember, non-sister chromatids cross with each other. I repeat, non-sister chromatids cross with each other. Dikinesis. Chromosomes regain their original structure after crossing over. Nuclear membrane and nucleolus begins to disappear. This marks the end of prophase 1. Point to remember, nuclear membrane and nucleolus begins to disappear. I repeat, nuclear membrane and nucleolus begins to disappear. Metaphase 1, the nuclear membrane and nucleolus disappear. The spindle fibers are produced. Chromosomes are arranged on the spindle fibers. Point to remember, chromosomes are arranged on the spindle fibers. I repeat, chromosomes are arranged on the spindle fibers. Below you can see different stages of meiosis 1. Next phase is anaphase 1. Chromosomes move towards opposite poles along with the spindle fibers. One chromosome from each pair separates and goes to opposite poles. The actual reduction takes place in this phase. Point to remember, chromosomes move towards opposite poles. Chromosomes move towards opposite poles. Telophase 1. Chromosomes reach opposite poles. Nuclear membrane and nucleolus reappear around each set of chromatids. Point to remember, nuclear membrane and nucleolus reappears around each set of chromatids. Cytokinesis 1 takes place by formation of cell plate in plant cells and furrow formation in animal cells. It is the division of cytoplasm. Due to this, one cell with 2 nuclei divides into 2 cells. Chromosomes begin to uncoil. Point to remember, division of cytoplasm. Division of cytoplasm. Meiosis 2. Prophase 2. Chromosomes become distinct and clear. Nuclear membrane and nucleolus begins to disappear in both the cells. Point to remember, nuclear membrane and nucleolus begins to disappear. I repeat, students, please remember, nuclear membrane and nucleolus begins to disappear. Metaphase 2. Nuclear membrane and nucleolus completely disappear. Spindle fibers appear in both the cells. Chromosomes are arranged on the equatorial plate of the spindle fibers. Very clearly you can see in the picture down that the chromosomes are arranged on the equatorial plate of the spindle fiber. Point to remember, chromosomes are arranged on the equatorial plate of the spindle fibers. Chromosomes are arranged on the equatorial plate of the spindle fibers. Anaphase 2. Chromatids move towards opposite poles along with the spindle fibers. Chromatids appear V-shaped. I repeat, point to remember, chromatids move towards opposite poles. Telophase 2. Chromatids reach opposite poles. Nuclear membrane and nucleolus begins to disappear, and when it reappears around each set of chromatids. Point to remember, nuclear membrane and nucleolus begins to reappear around each set of chromatids. Cytokinesis 2 is perpendicular to cytokinesis 1. It occurs by formation of cell plate. As a result, 4 daughter cells are produced with half the number of chromosomes as that of the parent cell. Point to remember, cytokinesis 2 is perpendicular or at 90 degrees angle to cytokinesis 1. In this picture you can see telophase 2 on the left hand side top, right hand side top telophase showing cytokinesis. This is the overview of all the stages of meiosis 1 and meiosis 2. A diagram is showing stages of meiosis 1. AC, prophase 1. AD, prophase completed. AD is metaphase. AE. Once again, metaphase. AF, anaphase, and AG, telophase. BH, prophase 2, BI, metaphase 2, BJ, anaphase 2, BK, telophase 2, and BL, cytokinesis 2. I repeat, C, interphase, D, prophase, E, metaphase, F, anaphase, G, telophase, H, prophase 2, I, metaphase 2, J, anaphase 2, K, telophase 2, and L, cytokinesis 2. Another view showing different stages of meiosis 1 and meiosis 2. Please observe carefully. I repeat in the order: leptotene, zygotene, pachytene, diploatine, dikinesis. This is prophase 1. Then metaphase 1, early anaphase 1, late anaphase 1, telophase 1, interphase 2, prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. Friends, we conclude with the significance of meiosis. It helps in maintaining a constant number of chromosomes over the generations. Through crossing over, new genetic material and combinations are produced. This leads to variation that provides the raw material to evolution. We end this presentation with the animated clipping of meiosis. Metaphase 2, anaphase, telophase, cytokinesis, meiosis 2 has started, anaphase 2, telophase 2, and cytokinesis 2. Thank you, students, for the presentation.